Hello, everyone, and thank you for joining us this evening for the second week of Rugby Research and Injury Prevention Group 2026 Journal Club. We will first begin our Journal Club presentation with our first intern, Hunter Zhu, from Lexington, Massachusetts. Hunter will be presenting on platelet-rich plasma and bone marrow-derived mesenchymal stem cells in sports medicine. Hunter, when you're ready, will you please share your screen?
Thank you, Elon, for the introduction. So, for those that don't know me, I'm Hunter. I'm currently finishing my junior year here at Lexington High School in Massachusetts, and for my article, I read a review article about platelet-rich plasma and bone marrow-derived mesenchymal stem cells in sports medicine. This was a review written by Alberto Gobi and Matthew Fishman in the Sports Medicine and Arthroscopy Review in 2016. So regenerative medicine is a fast-growing field of science and research, and it mainly focuses on regenerating, as seems, parts of the body for treatment. And platelet-rich plasma contains factors to help healing, and mesenchymal stem cells are pluripotent, meaning they have not differentiated yet and can go in follow different lineages. So, for example, in this image, we will be mainly focusing on their lineages in cartilage and bone. And this is very exciting for growing research as in the orthopedic profession people are studying and doing more research on directing lineage into specific directions for orthopedic treatments. Some key terms to know before this presentation are platelet-rich plasma, which will be abbreviated as PRP. This is plasma derived from centrifugation on a sample of the patient's own blood. Another important term is bone marrow aspirate concentrate, which will be abbreviated as BMAC. And this is bone marrow. This is harvested from the bone marrow and placed in a centrifuge like PRP to separate stem cells and healing proteins. And then finally, mesenchymal stem cells are cells found in bone marrow, adipose tissue, and umbilical cord blood that have the ability to differentiate into tissue, as mentioned before. The objectives of this review article were to understand the different applications of PRP and bone marrow-derived mesenchymal stem cells in orthopedic contexts. So specifically, they cover three general areas, which are soft tissue healing, which include examples like torn meniscuses and the rotator cuff. Another subtopic was cartilage defects, and finally, treatment in osteoarthritis, which is a condition in which the cartilage near the bone, shown right here, starts to degrade and can cause pinching feelings, pain, and lack of mobility. Um, and so, how are these concentrations derived? PRP comes from centrifugation of their patient's own blood, and as seen here, due to the difference in density of plasma, white blood cells or leukocytes, and platelets and red blood cells, when you spin them around in a centrifuge, they start to separate by density, and that's how you get the separation of the platelets. What's interesting is that this can create different concentrations in different studies, as studies have shown too much or too little concentration of platelets can negatively impact their effectiveness. And also, the same goes for leukocytes, as it's been shown that leukocyte poor plasma actually is slightly more efficient in these treatments. As for BMAC, these are osteogenic derived stem cells, meaning they come from the bone. A needle is inserted into the bone marrow and withdraws some blood, and also placed into a centrifuge. And non-hemopoietic precursors and platelets are isolated. In addition, they can be further expanded to increase the concentrations of mesenchymal stem cells by growing them in labs and culturing them. So here's like the kind of general trends that we're seeing with rich plasma in these studies. For soft tissue healing, there were limited results on rotator cuff tears in randomized control trials, and also there was signs of slightly decreased retear rate. However, there was minimal impact on function and satisfaction. As for cartilage defects, most studies compared control treatments such as scaffolding or collagen gel implants with PRP augmentations of the same treatment. And they generally showed greater cartilage healing with PRP treatment. Sinclair and colleagues found usage of augmented scaffolds led to significant improvements in outcome scores in patients that took PRP. However, there are studies that dispute the results, such as Kahn and colleagues, that either showed no difference in the treatments or actually showed that PRP treatment performed slightly worse. As for usage in to confront osteoarthritis, it showed that there was more improvement than placebo and hyaluronic acid injections, which are the typical treatments for osteoarthritis, which re that bring back some fluid to restore the shock absorbing capabilities around the joints. With PRP, it showed that there was improved Western Ontario and McMaster University Arthritis Index and also International Knee Documentation Committee scores. And these are two self-evaluation tests similar to the tests we learned about last week with ACLs. As for bone marrow derived MCS, in soft tissue healing, animal studies showed menisci healing with fibrocartilage compared to fibrous healing patterns in without MCS. Um, this is important because fibrocartilage is more similar to the native meniscus fibers, and because it has more structure and is more organized, it is better able to replicate um, the biomechanical usage of the meniscus fibers as they're stronger and less likely to re-tear. In human experiments with allogenic stem cells from other patients, it showed that there was an increase in meniscus volume, and this is important obviously because it increases the ability to cushion your joints and bones. In humans, there was also a decrease in re-tears and increase in healing of injured rotator cuffs. After 6 months, 100% of MSC tears healed compared to 67% without MSCs. And in a 10-year follow-up, there showed a 13% to compared to a 54% re-tear rate with MSC. Um, as for cartilage defects, noticed in rabbits that there was treatment with cultured chondrocytes and cultured MSCs had similar improved characteristics. With cultured chondrocytes, cultured cartilage cells will be replaced back into the injured part and regenerate slightly the cartilage. And this is important because it showed that MSCs had similar improvements when used as treatment. And in a study using BMAC and matrix-induced autologous chondrocyte implantation for patelliformal lesions, both groups significantly improved with complete filling in 81% of BMAC-treated patients. However, there was no significant difference. As for osteoarthritis, many studies show improvement in clinical scores, reduced subchondral bone edema, which are fluids in the bone that can cause pain, and increased cartilage healing. So, as for limitations, there are studies that show PRP usage on knee osteoarthritis have shown higher rates of adverse reaction. In addition, there are studies that dis that do not follow the same trend as other studies that say the usage of these treatments is not that show that the usage of these treatments is not conclusive. And thus, recommendations for future research in this would be more studies on safety and efficacy, as this review article did not cover any research covering the safety of PRP or BMAC. And also, the efficacy must be more studied because of the inconsistencies in data. As for medical implications, MSCs have potential to differentiate into certain cell lineages as covered before, and this is important in use of targeted therapy and regenerative medicine, as instead of replacing knees and joints with steel plates or replacement knees, we can try to use regenerative therapy to maintain the natural biological uses of our joints. As for applications for RRIPG, many of us want to go into orthopedics, so this is very important to understand the different treatments and new studies on PRP usage and BMAC usage for cartilage and tendon repair. And also, this is important for rugby because rugby, athletes are often in constant impact with each other because it's a very physical sport. And there's also very high volume training that can lead to meniscus tears or osteoarthritis. And it's important to understand the possible treatment methods that are currently being studied. This is my reference, and thank you so much for listening to my presentation. Any questions?
Thank you for that. I think what's kind of interesting about this area, as it's come very far, um, we were involved with quite a few early studies that had included many of the, as you kind of spoke about, some of the early, um, I guess, structures of how they were using this and implementation of it. Um, what I want to say is that, this is why, um, and it's not that I'm promoting it. What I'm saying is that there is a very large, you know, medical tourism going around this area. Some people want to say it's costs. Some people want to say they're going around lengthy clinical trial processes. You know, just obviously there's multiple factors of why people are going around doing this and going around the FDA structure. Um, Again, you hit it right in the nose towards the end of your conversation or presentation. That is, many regulations are challenged. It's very, very problematic. Um, and again, obviously there wouldn't be this medical tourism if there wasn't like a loophole to try to go around what's going on in the United States. But again, they have these systems in place to protect us. And if we don't, and we go around that to just try to get, stem cell therapy in some foreign country, it may not be most beneficial. They may not have the proper, care. And there's many, many horror stories about this all. But it's very well acknowledged that many people go overseas to get these treatments and care because of the benefits of them. So again, you did also speak about that. This has been well known to like really benefit athletes, whether they're how can I say collision athletes or whether they're impact athletes. You know, I guess what happens is that I guess the true long-term results are still out. It shows that there have been, improvements with this, but the problem comes down is that, we haven't seen the really long, exhausting long-term studies because there's so much skirting going on about it. And did you come across any literature on that, Hunter, or no?
Not really, but I do know I have heard a lot of stories in the media, in the news, of professional players going to get stem therapy, and I think it's very interesting that we hear about all these players that are recovering faster, coming back from ACL tears, Achilles tears, within shortened time periods because, and that they credit to stem cell treatment, and I think that's caused kind of a popularity or maybe a trend to kind of explore this mode of treatment. But also, like I said at the end of my presentation, it's also important to understand, um, obviously we don't know how these therapies are actually going on in the NFL, the MLB or whatever, and it's important to understand the risks and how effective it is for non-athletes in addition to athletes.
Very well said. Very well said. And like I said, it's not only athletes themselves. You have, people going to feel better. Maybe they have generalized arthritis, rheumatoid or variations of arthritis. Maybe some of them, have some varying disease, genetic diseases that induce, maybe an arthritis early on. The whole idea is that, it's known to make people feel better. So you're never going to be able to control that fully if there's a direct benefit, right? You're going to seek it out. You're going to look for that care if you know that it's there and somehow it's, and you're debilitated and you need to feel better, you're going to seek it out, right? Anyway, very good job, son. I know it's a hard topic and probably very political climate to be discussing this on, and I'm sure you probably you did a good job focusing on what you had to present because again, you could tangent off in all these things. They got I think Leon's did a paper on the critical review of international. You know what, Lily? Send that paper to Answorth, please. Leon's, I think, is a literature review, a systematic review, critical view on medical tourism, something of that nature. Just so he knows what's going on. Anyway, like I said, very good topic. Next, please. Anyone else have questions? It's really come a long way in the past decade from when, we were messing around with, cells and we are just basically trying to get them in matrices and trying to get them just to work. Now they've really got it down. So it's really beneficial for everyone. But again, many of them are in clinical trials. So many of them, you have to go to really high-end institutions and they're very costly.
Yes, thank you, Hunter, for that wonderful presentation, and thank you, Dr. Lopez, for the insight. Our next intern is Taya Maharaj from Stony Brook, New York. Taya will be presenting on the glymphatic system distribution as a mediator of brain trauma and chronic traumatic encephalopathy. Taya, when you're ready, will you please share your screen?
Okay, perfect. So thank you, Elon, for the introduction. My name is Taya, and I recently graduated from Stony Brook University, and I'll be presenting on the glymphatic system disruption as a mediator of brain trauma and chronic traumatic encephalopathy by Susan and her colleagues. This was published in a journal called Neuroscience and Biobehavioral Reviews in 2018. So to begin, for some background, first I'll address the glymphatic system, often abbreviated as GS. So basically, this is a lymphatic waste system located within the central nervous system. And because the brain is highly metabolically active, it produces a large amount of waste products. So they need to be removed efficiently. Simple diffusion alone is too slow to do this. So the brain relies on the glymphatic system to get rid of these substances. So the GS system relies on three main components. The first is the cerebrospinal fluid, which flows through the brain and helps flush away waste. Second is the interstitial fluid, which occupies spaces between brain cells and carries waste products toward the cerebrospinal fluid for removal. And lastly are the astrocytic end feet, which is specialized extensions of astrocytes that surround blood vessels. And these structures contain channels such as aquaporin 4 that facilitate fluid movement throughout the brain. And together, all of these components create a pathway for clearing metabolic waste and maintaining good brain health. For some additional background information, one of the primary functions of the glymphatic system is removing proteins produced during normal neural activity, such as beta amyloid and phosphorylated tau. And when these proteins aren't adequately cleared from the brain, they accumulate in the extracellular space and basically contribute to different issues such as acidosis, swelling, the development of diseases such as Alzheimer's, and in our case, particularly relevant to us, CTE. So an important aspect of GS function is that it works most effectively when we're sleeping as opposed to when we're awake. So when we're sleeping, the interstitial space expands, allowing significantly greater movement of CSF and ISF through the brain tissue. And this increased fluid exchange dramatically improves the removal of metabolic waste. But traumatic brain injury, or TBI, significantly disrupts this process. So after brain injury, the brain would undergo what's known as a metabolic cascade. And this is basically where cell membranes become damaged and release excessive amounts of glutamate, which creates a lot of metabolic instability. And as a result of this, large amounts of protein-rich waste rapidly accumulate in the extracellular space, which adds an additional burden on the injury, on the glymphatic system that may already be impaired because of the injury. So with this study, the purpose was to to evaluate the relationship between mild traumatic brain injury and glymphatic dysfunction, and to determine how these changes may contribute to the development of CTE. And the authors proposed that sleep disruption following an injury may serve as a key mechanism during waste accumulation, chronic inflammation, and long-term neurodegeneration. So on this slide is the graphical abstract that was from the paper, and it basically illustrates the author's proposed model showing how brain trauma leads to both a metabolic cascade and acute glymphatic disruption. So these processes together contribute to sleep disturbances, which then worsen waste accumulation, as I've been saying, and increase the risk of neurodegenerative disease over time. So as for the methods of this study, it was mostly a comprehensive literature review rather than an experimental study. So the authors analyzed findings across multiple levels of TBI severity, with particular emphasis on mild TBI and repetitive head trauma. They also examined the role of insomnia because sleep disturbances are extremely common following TBI. So to support their conclusions, they also integrated evidence from different sources, such as human autopsy studies and established models such as the hit and run mouse model, sorry. And this approach allowed them to connect their findings from cellular, animal, and human research. As for the results of the study, they had several important findings, first of which include that TBI disrupts aquaporin 4 channel function. So after injury, aquaporin 4 channels become displaced from the astrocytic end feet where they normally facilitate fluid movement. So because of this displacement, these channels that are usually essential for glymphatic flow, they are significantly reducing the brain's ability to clear waste products. Additionally, sleep disturbances play a huge role. So up to 80% of individuals with mild TBI experience sleep-related problems. So since glymphatic clearance occurs primarily during sleep as opposed to when we're awake, as I mentioned earlier, ongoing sleep disruption prevents the removal of those proteins, the beta amyloid and phosphorylated tau, which can lead to chronic accumulation of these toxic proteins. And another key insight is that the authors found an important anatomical connection between glymphatic function and CTE. So in CTE, phosphorylated tau protein accumulates heavily in perivascular spaces, which are the same regions that serve major drainage pathways for the glymphatic system. So this overlap between the two systems suggests that repeated trauma may damage the brain's waste removal system and can contribute to neurodegeneration. So for the discussion, the authors propose that glymphatic dysfunction may help explain how repetitive head trauma progresses into CTE. So after a single injury, there's an opportunity for recovery. Adequate rest and treatment can support waste clearance and allow glymphatic function to normalize over time. However, repetitive injuries repeatedly trigger metabolic cascades, resulting in persistent waste accumulation and ongoing disruption of the glymphatic flow waste removal system. So importantly, the neurofibrillary tangles, which is basically an accumulation of the hyperphosphorylated tau protein, are attributed to CTE and often found in the areas that have the greatest glymphatic activity, such as the perivascular spaces and the glia limitans area of the brain. So in simple terms, this basically means that the phosphorylated tau accumulates in the regions the brain normally uses to clear out waste. So when trauma damages these pathways, tau clearance becomes less effective, and this leads to protein buildup and permanent brain damage. Therefore, the authors suggest that failure of the brain's natural cleaning system may be a key mechanism linking repetitive head impacts to the development of CTE. As for some limitations with the studies, the first is that there are important differences between human and animal models. So species such as mice, rats, and sheep lack some of the complex drainage structures found in the human brain, which may limit how well this findings translate to humans. Secondly, researchers currently lack validated biomarkers that can measure glymphatic flow or phosphorylated tau accumulation in living patients. So this makes it difficult to track the disease progression in real time. And similarly, much of what we know about CTE comes from postmortem autopsy studies, so which basically means that the patients being studied are dead. So because researchers examine the brain only after death, it's challenging to observe how the disease develops in real time. So a few key takeaways from this study is that sleep is not only restorative, but it is a critical biological process that allows the brain to clear potentially toxic waste products. Next, the TBI creates a harmful feedback loop. So trauma damages the glymphatic structures and basically the cleaning system of waste products in our brain, while also causing sleep disturbances that further impact the waste clearance system. And lastly, sleep may represent an important intervention target. So improving sleep quality after injury is very important and can potentially reduce long-term accumulation of these toxic neuronal proteins and lower the risk of developing CTE down the line. That is the end of my presentation, and this is the reference for the article that I used, and now I'll open the floor to any questions from any interns or Dr. Lopez.
All right, well, I'll start it off. What happens is a very, very complex topic. Thank you so much, Taya. It was very hard, to kind of present that topic. So you did a very good job doing that. Basically, as she concluded, try to understand the whole glymphatic system and what its purpose is. And that is, obviously, if you have disturbances, other conditions associated with like problems with it, headache disorders, migraines, mood disorders, anything with the brain, it affects your brain from clearing out the toxins or probably going into REM. These are folks who have difficulty reaching that capacity. And that's why, it's a fine line of when people get a traumatic brain injury, we want them to stay awake, watch out, see how they're doing, because we want to make sure the brain has attempted to clear out or has drained itself of maybe some of the injury. You can kind of see what I'm saying in the simple form, right? If you have a traumatic brain injury or a concussion or something like that, and they say, oh, don't go to sleep, don't go to sleep right away, stay awake. We want to make sure, just rest your eyes, but try not to go to sleep, is because they want your system to try to reboot. And they want particularly your lymphatic system, to get back to normal pressures, so you don't have sleep disorders, you don't, encounter, long-term traumatic brain injury with the deposition of tau, things of that nature. It is an important system to understand. So thank you, Ty, for bringing it up. But like I said, it's very challenging to truly understand the micro or the molecular biology that goes on there. So next time, a little bit more pictures to show the blood-brain barrier, maybe to show how the materials are passing back and forth, things of that nature. Really puts it in perspective, so this way people understand what it is. Okay, everyone? So some of the traditional images can show the glymphatic system. Let me just grab this image real here real quick, see if it allows me to. So as you could see, this is your glymphatic system, right? And as you could see, here you are, some of these molecular molecular products, young Miss Taya presented. And here's your waste that they're talking about. It lies in between. So when you get it out of your periarterial space, right, it comes in through the arteries and then exits at the venous system. So you're looking at it, the arrow is the flow. See, cerebral spinal fluid and going in through the artery, your blood-brain barrier, protected, astrocytes, and then coming across to your venous system and then coming back out. And this is where the waste, these little dots are your waste, where they line up here. And then obviously, you don't want anything to remain there. And that's really kind of, this helps everyone understand it just a teeny bit better, right, guys? What do you think? Right? Just just a little image just so you understand if we're looking at the blood flow, right? Because there is no blood in the brain, it's cerebral spinal fluid, yes? For those at the, learning level, at the beginning level. So what it is that you could see is glymphatic system. That's what we're talking about here. All right, everyone? So maybe we try to put it a little bit more in perspective so you could see the materials and then the molecular biology of it. All right? And like I said, sometimes it's a little easier when you just look at it. But what young Miss Ty brought up was, the importance of the system functioning well. Because if it's not, right, migraines, headaches, sleep disorders, everything you could possibly think of, and let alone if you have a traumatic brain injury inducing that. Okay? Any questions on that? All right, thank you.
All right, thank you for that. Our last and final intern will be Jason Tay from Massachusetts. Jason will be presenting on harnessing an AI-driven analytics model to optimize training and treatment in physical education for sports injury prevention. Jason, when you're ready, will you please share your screen?
All right, thank you so much for the introduction, Elon. Hi everyone, my name is Jason Tay. I'm a rising junior at St. Mark's School in Southern Massachusetts. Today I'll be presenting a 2024 paper from the ICEMT conference, Tokyo, Japan, and it's titled Harnessing an AI-Driven Analytics Model to Optimize Training and Treatment in Physical Education for Sports Injury Prevention, and it's by, sorry if I mispronounce anything, but Nilamata Mitchell and colleagues. Okay, so to set the stage, sports injuries, as we know, are a major problem. They can threaten athletes' careers and they can create real financial burden for teams and organizations. And so the author's argument here is that traditional and human-based PE and physical training designed to teach students about biomechanics and how to stay healthy, they can be subjective and generalized. For example, if a school chooses to a certain PE program, they're going to stick by that regardless of an athlete's certain needs. And so as a result, this can fail to address every need for every athlete, and this can fail to prevent injuries. So the goal here is to develop an AI-based prediction model that can identify injury risk patterns from continuous monitoring of health data, and ideally it's going to flag risks and any patterns that can lead to injury before they actually occur. And just a quick note for this technology, this paper is going to use called a neural network. And so what a neural network is, it's kind of a loose computational model of the human brain. And so it's going to take data and the input, as you can see in the figure, and it's going to pass it through these layers of interconnected nodes. And what each node does, it has a set of like variables, which are called weights. And you're going to tune the data set in order to really match the pattern the data gives. And overall, it's going to learn data on its own, produce its own pattern. And so before the old model, the authors reviewed this in the landscape. One piece of prior work uses what's called a dual fusion structure, and it's basically a two-pathway design. And so with neural networks, the most basic part of neural networks is that they use one pathway. And an issue with that is the one pathway case, the model is just trying to look for the pattern itself, and in the process, it can maybe forget or leave out the raw details. And how the dual fusion structure solves this is by incorporating two pathways. So one's trying to get down and find the pattern, and the other one preserves raw data. And by combining these two, the model can really get to the next level in classification. And as you can see here in the cited paper, at an accuracy of up to 97%. The most important concept that the paper uses is the recurrent neural network, or an RNN. It's a type of neural network. And so the key distinction about an RNN is that it's built for time sequential data. Unlike a regular neural network, which looks at a single snapshot and forgets it, an RNN has a form of memory. The output from each step, as you can see in the figure, feeds back as part of the next input, as you can see per arrow. And so it's going to look at how an athlete's data evolves day after day. For example, a single day of athlete just high intensity work, it just doesn't mean much to a regular neural network. But a recurrent neural network is going to look at the entire timeline. So maybe multiple weeks of high intensity training with maybe patterns such as low sleep, poor nutrition, poor hydration. This sort of patterns look at flags for injury risk. They also cited prior work applying RNNs to spinal cord injury classification, confirming the use of RNNs in this context, as well as work on wearable AI devices that monitor an athlete's biomechanics for data collection. And so we're now moving to methodology. And so we can see our framework here. The data is going to come from the continuous health monitoring of each athlete per the sensors. And so for the methodology, they describe four functional pillars. You can see here they're PT and injury prevention. Second is training the RNN for injury protection. Then forecasting injuries with RNN and implementing precautionary measures based on the model's output, like remain and rest. As you can put a cycle diagram, it's all kind of a loop. So we're going to start with physical training and ensuring that the data is being collected and the athletes know through the movements. And then it's going to next move on to training the RNN based on the data and try training it to make sure that it can predict injury risk. And the third part is analyzing predicting. So it's going to take the model, it's going to take new data perhaps from another athlete, and so you to actually predict injury risk, or maybe the same athlete, and just kind of get to a conclusion. And from there, with kind of output, it's going to be corroborated by medical officials and maybe peer reviewed in order to ensure that it's true. And then it's going to be sent over to the medical officials to maybe put in precautionary measures for the athlete. And this is going to loop back to physical training because the new measure going to be implemented maybe in their plan or the PT plan or the nutrition. And this is going to be a form of full cycle. Okay, so for the technical details, just a few. So this they use what's called a time distance modeling, which is analyzing not just what changes in data, but the rate and the trajectory of the change. So for example, a heart rate of 70 in an athlete is just there. It's not really a remarkable data point. But maybe a heart rate that's been climbing by like 5 beats a day for a week, or like maybe nutrition that's been like going down, or caloric intake that's been going down for multiple days, it tells a very different story. And so for comparison, we have two comparators, and one is going to be a support vector machine or an SVM. As you can see in the figure to the right over here, it's just basically a mathematical model that just like sets the most mathematically clean boundaries, just like straight linear cuts. And then the other one's called a placement model, so it's kind of the opposite, as you can see on the bottom right, where it's more of a range of values rather than clean cuts. And these two just provide a good span to test the RNN on. One thing to note here is that despite the article mentioning actual professional athletes, the data set, or the model, sorry, were tested on fourth and eighth graders, so data is from fourth and eighth grade PE students. And so now we're going to get to our results. So they tested on three main metrics, and the first one is assessing the effectiveness of treatment for injury prevention, which is essentially just seeing whether or not a certain PT or certain protocol can really help prevent injury. And so we can see that the RNN model improved consistently over time. Eventually went from 85% to start, but eventually went to 99% accuracy, beating SVM by 9% and like by 18%. The second metric is the impact of PT and treatment on injuries. So it's just seeing whether or not certain movements can maybe overload the athlete and lead to higher injury risk. Here, the RNN continues to outperform both comparative models and even the human-based expert score by significant margins, as you can see per the numbers. And then for the third metric, it was a little more distinct, but it's general P training identification. So just identifying movements probably for application in the other two. And we can see here it excelled in accuracy and performed reasonably well in precision and recall, with scores in 99% accuracy and in the low 90s and high 80s for precision and recall. So when it comes to interpreting results, we can see that RNNs can be strong in separating and interpreting visual data for posture and movement assessment, as well as classifying maybe movements for injury prevention. They suggest a few practical applications, a feedback tool for PE educators, fine-tuned training regimens maybe that account for injury risks, and AI development of personalized treatment plans to optimize recovery. So kind of vision they're putting forward here is kind of fairly ambitious. However, like any paper, there are some limitations worth noting. And being clear about these just helps us judge how applicable the findings really are. So first of all, the methodology and the architecture they describe is very vague. They introduce a lot of new terms all in one paragraph, and they don't really take the time to define it, which results in a very like vague architecture. And especially for AI model, it needs to have the good reproducibility, which this article fails to do. Second of all, as I have mentioned before, the work was conducted on students rather than professional athletes, despite mentioning them in the scope. And to be honest, athletes in this level are the most relevant population for this kind of injury prevention, so I feel like including them would have been beneficial. And then third, more technically on the results themselves, the paper never confirms what's called a train test split being used. And so in AI, just to summarize, a train test split is kind of defining where an AI stops training and starts testing. So a trained data set is what the model actually takes in for training to actually develop what it is. And the test data set is what it's kind of being evaluated on, like a test, like a real life physical test. The problem is the paper never tells you whether or not a train or test data set was used. And so it's more likely than not that, as we saw, the RNN improving in the aspect, that was likely due to just the train data set being used over and over again rather than the new test data set. And this is a problem because it can indicate what's called overfitting, in which the model just like learns the same data set too well and it can't really make other predictions off of new data sets, which would essentially render the model useless. And finally, the results they give are presented without a confusion matrix or confidence intervals, either just to ensure that we can identify the model's failures as well as successes. For example, in injury prevention, what's called a false negative can be really detrimental because, say, for example, an athlete is actually injured and needs help, but the model flags it as, for example, healthy. This can be really negative because the athlete can then push through the injury and cause even more serious injury, put the athlete at further risk. And also, accuracy alone is a flawed metric because despite them citing 90% accuracy, there are some cases in which maybe it's an imbalanced data set, and so maybe there's a lot more athletes who come out as healthy than injured. Then a model can predict maybe every athlete is healthy and still get a high accuracy because because the data set is flawed in that way. But in conclusion, we can note that AI and RNN models can continuously monitor injury dynamics and support personalized, data-informed injury prevention. And that despite the article kind of being vague about the application, we can note that it still has potential based on the RNN's capabilities. The article cites a few future works, including the integration of AI, the sort of AI analytics combined with real-time sensors for injury prevention, maybe on the court or on the field. They also mentioned genetic profiling combined with sort of model to test injury accessibility, as well as using VR simulations for athlete decision-making training. And to bring it back to why it matters for us RRIPG, the sort of predictive modeling can be really helpful, especially in sports such as rugby, where there's higher injury risk or maybe the injuries are a more significant degree. And thus, using this model to maybe optimize athlete habits and maximize the recovery can really be a great way to facilitate injury prevention and making sure athletes stay healthy. Here are my references, and that's going to be it. Thank you so much.
Thank you so much, Jason. Jason, I'll open the floor to any interns or Dr. Lopez if you'd like to add anything.
Yeah. All right. Well, thank you so much, Jason. That was a phenomenal talk. Very high level. Obviously, everyone's still trying to land from your presentation. The important thing is, I guess, if we can go to the last slide, the implications of what you were saying of integration of the analytics of biomechanical sensors. This is something that they've been really, really trying to understand better. Did the article actually state that they want to do this?
Yeah, that was part of the future works, as I mentioned.
They did speak it right.
Yeah.
So what happens is that if you have the biomechanical sensors, you have exact data, but then you have artificial data. It gets a little tricky because you're now using artificial intelligence to evaluate. You can't assume that all data points are specific. They may be sensitive, but they may not be specific. I think, Jason, everyone, what I'm trying to tell you is that, when the artificial intelligence takes the data and it tries to manipulate it, I can only assume that it's making assumptions. But meanwhile, if you incorporate the biomechanical data, which are exact data points, I really wonder where that clash is of limitations in the data analysis overall. And that's really kind of something that maybe, Jason, you can did you find anything when they were talking about that or no?
There wasn't a lot on it, but I'd probably say like AI is still a pretty like developing topic, and it's just a matter of whether or not they can optimize the models well enough for this application. I'd probably say it would
Sure.
take a good while before they could probably get a model that can successfully carry this out with like minimal error.
This is exactly why when we build our models, and again, the way I'm speaking, Jason's speaking right now, give it some time, everyone. Keep reading. You'll learn about what we're talking about here. When we develop our models, for our analyses, what data points do we incorporate? Do we incorporate, descriptives, demographics, age, playing level, BMI, are these descriptives, demographics influential upon the model in producing a statistical relevance? And then from that statistical relevance, then you can coordinate the statistical relevance with the biomechanical markers to try to see if you can come up with a more valid outcome or understanding. And that's really kind of a simpler way of trying to explain that. So if you have a good model, and this is why, a lot of papers that use data sets that are unvalidated, and don't get me wrong, I mean, if you want to use a data set to hypothesize, maybe test something, that's fine. Maybe even test a model, test your proposed model. You can do that. There are data sets, there are systems that are available, and you can get full sports data sets. But the question comes down, is that data set really valid? Is it, captured appropriately? Does it follow the definitions? And that's really where it gets a little tricky in the bigger picture of things. So on that note, I won't go too much into it because we still have a lot to learn on this, but very good there, Jason. Very good.
All right, thank you so much.
Yeah.
Yes, thank you, Jason, and thank you, Dr. Lopez. This will conclude the second week of the Rugby Research This will conclude the second week of the Rugby Research and Injury Prevention Group Journal Club. Thank you all for attending.